Have you ever wondered how your early stem cells knew which cells they would graduate to become? Whether they'd be a bone cell or a muscle cell. But first, what makes a bone cell different from a muscle cell? In simplest terms, it's the proteins they produce, which are coded for by genes. But how can these cells produce different proteins if they all have the exact same DNA? Chromatin regulation. But let's ask the expert. Let's take it to the protein synthesis restaurant. Hi there, I'm the chromatin regulation chef, and on today's menu, we've got DNA methylated heterochromatin and histone acetylated euchromatin. English, please. The heterochromatin's more like angel hair pasta, and the euchromatin's more like fettuccine. In DNA methylation, we start with our chromatin. Then we add in our spices, sprinkling in methyl groups and DNMT enzymes to speed up our cooking. When cooking, the methyl groups attach to the cytosine bases in the DNA. When it pops out, we get a tightly packed chromatin dish, much like this angel hair pasta. Pretending our fork is RNA polymerase, it's difficult to grab that single desired strand or gene. So we can't transcribe the gene, can't synthesize our protein. Zooming out, that means that certain genes are not expressed in that cell. So pretending that that chef feeds the bone cell community, she's DNA methylating the muscle-specific genes because you don't need those in a bone cell, differentiating the two cells. But what about the genes we do want? In histone acetylation, we still start with our chromatin, just remembering our histones. We add in our spices, but different ones, acetyl groups and HAT enzymes. When cooking, you gotta remember that your histones in DNA have opposite charges. That's why they bind. They're dating. But the acetyl group binds to the histone, neutralizing that positive charge. It's like the histone cheats with the acetyl group on the DNA. So the DNA and the histone are less attracted. When it pops out, we get loosely packed chromatin. Like fettuccine, it's relatively easy for our RNA polymerase to swoop in, start transcribing, and synthesize our protein. Zooming out, that means that other genes are expressed in the cell. So pretending that she's a bone cell chef, she's histone acetylating the bone-specific genes because you really do need those there. And that's a wrap, or a pasta dish.